Hello, everyone. Thanks for coming to this talk. This is on a study that we did on the genus Reynoutria, Japanese knotweeds and relatives in Wisconsin. So knotweeds are in this genus Reynoutria, and there are two species and a hybrid between them, and both are, or all three of these are pretty widespread in the full invasive range. So that includes Europe and a lot of North America. The two species, the original parental species, are shown here: the giant knotweed, which you can guess why it earned its name, and then the Japanese knotweed. And one feature stands out on these images. One of the more useful characteristics for telling them apart in the field is the leaf base. So the blade base has either these lobes that point backwards in the giant knotweed, or it is fully truncate in a rather spectacular fashion, I think, in the Japanese knotweed. And it's been known for some time that there are fairly extensive hybrids, introgressed, offspring of hybrids, and so on. So there's a bit of a genetic mess to work out for these species. And up to this point, I think the naming has maybe gotten in the way a little bit. We have these two names for the original species, and then we have a name for the hybrid between them, this Reynoutria bohemica, the Bohemian knotweed. And it looks like even before we got our data, there's lots of evidence that the Bohemian knotweed is not only one thing. So having a name for this hybrid swarm of hybrids and introgressed offspring might not be the most correct way to encapsulate the diversity of this group. So these are herbaceous plants. They're rather large herbaceous plants, and they are good competitors and difficult to get rid of. So they're definitely on a lot of people's radar for a plant worthy of concern and possible eradication. So just on this image here, I've got the static maps on the right of their current distributions, and then on the left, synchronized maps of their spread according to herbarium specimens. This is from the GBIF database, so just grabbed the specimen data and then lumped it into 10-year increments and show the distribution of specimens that are known about. So a little bit of an advance in Europe, advanced spread there, and then North America, moving in from both coasts, really, all the accessible locations that this plant can grow. And the names on these will be, of course, subject perhaps to interpretation, but we have the definitely categories of Japanese knotweed, giant knotweed, and Bohemian knotweed here. Their native range is actually rather small. So Eastern Asia, all three species, in fact, the whole genus is distributed in the region that's shown in the green and the map on the right. And the names kind of tell you where they're a little bit more represented. So Japanese knotweed gets its name from Japan, of course. The giant knotweed has its name Reynoutria sakhalinensis, and this is Sakhalin Island here, just north of Japan. So pretty well distributed in that region, also in mainland Eurasia, neighboring to these islands. And a nice image of the flowers there. So sometimes if you look at even recent literature, they're not always classified in this genus Reynoutria. In fact, even in my lifetime, I've known them in 3 different genera, and this paper I think really solidifies what they should be called. So they spent time in Polygonum, and you can see that synonym name there. They're Polygonum cuspidatum. They've been in the genus Falopia. Falopia japonica was the Japanese knotweed in my experience for a while. But this study by Schuster et al. 2011 really puts Reynoutria off on its own, all the species represented here, and distinct from Polygonum, distinct from Falopia, distinct from Muhlenbeckia. So pretty clear classification there. And these are the giant knotweeds. So they don't really have their own common name for the genus, but they're the big ones. They have that shared habit of being taller than a person. So prior to our study, there were pretty extensive data, thanks to this group, Park et al., that studied plants mostly in their native ranges. So representatives from the islands of Japan, and then also Sakhalin Island, also mainland Korean peninsula, little bits of Russia and so on. And I've highlighted on here the 2 major clades, looking at the Plastid group and also the nuclear data. The blue is going to stand in this study, in this presentation, for the Japanese knotweed, and the brownish is going to stand in for the giant knotweed. And I've highlighted on here some of these non-native populations. So RS for Russia, United States, UK, United Kingdom, United States. And the paper that published these kind of downplayed, it seemed, this result a little bit. The relationship of some of these species actually having a hybrid signature, some of them with bits of Japanese knotweed and bits of giant knotweed mixed together using the nuclear data. So we took on this project and we wanted to expand on this sampling over a little more fine-grained sampling throughout the state of Wisconsin, investigating both the Plastid data and also the nuclear data with this LEAFY second intron locus. So getting the data back from the Plastid, we used only this matK gene, and there's not a whole lot of surprises here. As you might expect, the Plastid DNA more stable, and so we got a clean signature for Sakhalinensis type, the Japanese knotweed, and this big S here, and then one clean version of Japonica type, so clear types of Plastid in the Japanese knotweed and giant knotweed respectively. Sort of thankfully, right? This is something that has been used to identify these plants in the past, and as you might guess, the Plastid is not really a comprehensive identification for species, as we will see. So here's the mess that you get when you introduce the nuclear data, and this is much more complicated than I think people might have suspected, or for an invasive species, there's a lot of genetic diversity here. And so these are the genotypes. We put them into categories of similarity, so these monophyletic genotypes. And a lot of these are shared with one individual having more than one of these genotypes. So I put up an image of cytotypes, these karyotypes of various knotweed species. They're well known to be polyploid. Polyploidy is consistent with individuals being hybrids. So if they have a hybrid complement of a Japanese knotweed parent and a giant knotweed parent, that can contribute to polyploidization. And so just about every plant we've found has a mix of at least 2 leafy sequences. And for the majority of what we found in Wisconsin, they were a mix of Japanese knotweed and giant knotweed types. So these brackets here in the solid lines show the combinations that we found. S1 with J2, S1 with J1, and so on. So a Sakhalinensis or giant knotweed type mixed with a Japanese knotweed type. Several kinds of hybrids though, so not just one hybrid entity here. And it gets to be a little bit challenging to lay this out graphically. So we have on here, the squares I've shown are genotypes that are postulated. They may not exist in individuals. It could be that the parents were already rather genetically diverse. And then the circles with the pie charts on them, those show the genotypes that we found. The top of the pie is the Plastid genotype. So we have one here with a Sakhalinensis Plastid type. And the bottom would show the nuclear leafy genotype, either one color if it's one kind, or up to 3 colors if we've got 3 different kinds of leafy sequence in those. So we see some combinations that are this one only 1 leafy sequence, and that tracks to Sakhalinensis. Up here we've got 3 types of leafy sequence, but they all track to Japonica. And then down at the bottom, most of these, as you'll see, have a combination of blue and brownish. Bluish and brownish is Japonica type and Sakhalinensis type throughout a few different combinations there. Again, just looking at one state in the United States, so state of Wisconsin, lots of combinations. Morphologically, I haven't found a good, clean signal for any of these genotypes. There's some morphology that goes along with the parents. We have this one individual of what looks to be clean giant knotweed here in the orange, showing with at least one individual the multicellular hairs that are distinctive on the leaf underside, and then this deeply cordate leaf blade base. So pretty good, clean giant knotweed. And then over here, the closest thing we've gotten to a pure Japonica also has the morphology that goes along with Japonica. And then lots of the other ones, we found different genetic combinations and also lots of variation in the so-called diagnostic morphological traits. So I feel like we've uncovered something that's going to need a lot more attention here. We did find the ability with the LEAFY genotype to be able to identify these with a simple PCR assay. So taking advantage of a big indel insertion deletion site between Japonica and Sakalensis, we developed unique primers that could amplify one or the other of those, and this is just showing the success. Most individuals have both kinds of Japonica type and a Sakalensis type, and some individuals, this is a Japonica only, and this individual is a Sakalensis only. So this would be a quick way to identify these plants as hybrids or parental species using PCR. Geographically, a little bit of a pattern. You see the similarity maybe in some of the colors. In the sampling that we did was southeastern Wisconsin, northwestern Wisconsin, and then far northern Wisconsin. A little bit of pattern, not enough, I'd say, to really draw a firm conclusion with at this point. So moving ahead, we've got a plan to look for better markers, additional markers that might be able to tease out those individual genetic elements using PCR. Up to this point, we've been using the second intron of LEAFY, and we're going to try to explore some of the more variable parts of the LEAFY gene, both exons and introns. And lots of thanks to all the people who made this possible. I had lots of help from undergraduate students at UW-Whitewater, and a lot of help from the Wisconsin DNR and people who were working with them to send us samples. So very grateful for all the help we got here. And thank you for listening.